Assalamualaikum viewers, welcome to Academia Coaches. In my tutorial lecture, I will explain about epigenetic modifications. There are two types of epigenetic modifications: the modifications of DNA and the modification of histone proteins. Histone proteins is present in the form of octamer having repeated subunits H2A, H2B, H3 and H4. These are the tails of histone proteins which are known as histone tails. DNA is a double helical structure which have nucleotide bases cytosine, guanine, adenine and thymine. The modification of DNA is known as methylation. In the process of methylation, methyl group is directly attached to cytosine. In the structure of DNA, cytosine is always present followed by guanine, and this site is known as CpG site. Methyl group is attached to cytosine at CpG site. This is the structure of cytosine. The methyl group is attached to cytosine at 5th carbon position, which is also known as 5-methylated carbon or 5-MCO5-methyl cytosine. The methyl group is attached with 5-carbon cytosine with the help of DNA methyl transferases. When the methyl group linked with cytosine, it causes the silencing of genes. Silencing of genes. As a result of methyl linkage with the cytosine, gene is silenced and it causes mutation in which gene is not expressed and not transcribed, and the methylation modification stop translation or transcription of genes. The second type of modification is histone modification. There are two types of histone modification. First is methylation. In methylation, methyl group is linked with the tail of histone protein. And the second type of modification is acetylation, in which acetyl is added to the histone tail. It is known as acetylation. The acetylation of histone protein is performed by histone acetyl transferase. Histone acetyl transferase add acetyl group to the lysine amino acid residue which is present in the tail of histone protein. Acetylation of histone protein causes activation of gene. The acetylation modification can be reversed by the enzyme histone deacetylase. Histone deacetylases remove acetyl from the lysine residue, and it results in gene deactivation. Do not forget to subscribe my channel for latest and more relevant videos. Thank you.